Hi everyone, it's Mrs. Bustamante. Today we're going to talk about 2.5, which has to do with the cell membrane and its permeability. So last time we were together, we were talking about the cell membrane and how it was a fluid mosaic. We talked about it being fluid because it's made of these phospholipids, right? And they have a polar head and a hydrophobic tail on them. So we talked about proteins that could go through them, right? Some proteins, transmembrane proteins, would need both a hydrophobic and a hydrophilic part. Some of them, if they were just a peripheral protein, right, would only need that hydrophilic part on them. And then we talked about some glycoproteins and some glycolipids. And if it was a glycoprotein, right, it had this carbohydrate chain coming off a peripheral protein. But if it was a glycolipid, it would just come off of this lipid. And a lot of times those are kind of like the fingerprints, if you will, of the cell. Those are going to be used a lot in communication and identification with the cell and other cells itself. Something else we briefly talked about was cholesterol, right? And how cholesterol helps regulate the cell membrane, make sure it doesn't get too melty or melt altogether if the temperature gets too hot, or freeze if the temperature gets too cold. All of those things, because that membrane, the cell membrane, has all those different components, that's why we call it a mosaic, all these different pieces that get put together. And all these different pieces play some more important roles. They also have a function, right? We talk a lot about structure and function, and you'll notice when you look at these that they are structured differently. They look different, and that's because they have different functions, right? These carbohydrate chains on your glycolipids and glycoproteins are used for identification. Some of these are going to be used channel proteins for moving things in or out. And peripheral proteins can have some different functions as well as our function of cholesterol that we talked about. So what does that mean? Well, when we look at our cell membrane and we see all these phospholipids kind of lined up, we know that only some things can fit through because of part of it being polar and part of it being nonpolar. That's why we say the cell membrane is selectively permeable. So only some things, the selective part and permeable, can move through. What can typically move through pretty easily? Well, small nonpolar or non-charged molecules can typically move right through the cell membrane. They can kind of sneak between that phospholipid bilayer, those phospholipids, from one side to another. Typically, they're going to move from your high concentration to your low concentration, so we could describe it as diffusion, right? What are some of the molecules specifically that can do that? Gases, nitrogen gas, oxygen gas, carbon dioxide gas, all of those typically can move straight through the membrane, again, from a high concentration to a low concentration. So how do these larger hydrophilic or polar molecules or even ions, how do they get in and out of the cell? Because they need to do that as well. They need to move from one location to another. They can't move freely. They're too big and they're charged. We have these hydrophobic tails, so they're not just going to be able to fit through. So we have to use something called transport proteins. And there are two main types of transport proteins that we need to know. The first is a channel protein, and it looks just like this. It looks kind of like a big tunnel. It's a transmembrane protein, meaning it goes all the way through your phospholipid bilayer, and molecules can move through it. It is important to note, however, that only very specific molecules can move through. It's very specialized. It's not just going to let anything move through the phospholipid bilayer. Another type is this carrier protein, and a carrier protein is a transmembrane protein as well, and it changes shape to move the target protein through. We're going to talk a lot about a carrier protein when we talk about sodium-potassium pumps. And sometimes they require ATP, they require energy in order for carrier proteins to work, because they will often move things against the concentration gradient, meaning that they'll move something from a low concentration to a high concentration. And small polar molecules like water, which is just H2O, we think about it as kind of small. Well, some water molecules can actually pass directly through the cell membrane because they are small. But when we think about water, we talk about it being polar, right? And we know that a huge part of the cell membrane is actually hydrophobic. It doesn't like water. So not too much water can actually fit through here. And there's going to be a special type of protein that can move water through called an aquaporin that we will talk about later. One thing I always think is kind of interesting is we know that animal cells, the outermost layer of them is just the cell membrane, right? But plant cells also have this thing called the cell wall around them, this boundary that provides shape and protection from the cell. It allows it to keep from breaking open, essentially, if water pressure gets too high. And it helps the cells stand up against the force of gravity, right? All these things are really important for the cell wall. So how do things get in and out of plant cells? Well, we know they have a cell membrane, so that's going to function the same way, right? But they also have this rigid structure. So what happens there is what I like. So when we're talking about the cell wall is made of complex carbohydrates. In plants, it's cellulose. In fungi, it's something called chitin. All different things, but they're pretty complex and they're not easy to break down. So plants have kind of something special in them. Plasmodesmata. Plasmodesmata is in plant cells, and it's really just holes between plant cells that allow nutrients and waste to transfer in and out. You can see them here in this picture. Here's your plasmodesmata, and it kind of allows things to go in and out. So essentially it just has another little hole that will allow for things to permeate the cell, or rather out of the cell one way or another. I hope this was helpful in solidifying why the cell membrane is a fluid mosaic and how some substances can move in and out of the cell. We'll spend more time on this as we move through the unit, specifically about active and passive transport and diffusion of things in and out of the cell. Thank you.